Welcome to this introduction of ReadStore, a genomics data platform that allows your team to focus more on analyzing data and generating insights instead of managing FASTQ files and metadata. My name is Jonathan. I'm going to walk you through this session. ReadStore is super easy to set up. You can simply install the Python package, start a server, and connect to the web app. It can be deployed in any Linux server or on your private cloud and allows you to keep your sequencing data stored locally and in a compliant manner. Let's have a look at the user interface. We can easily upload FASTQ files from the command line or import files in the staging area. The FASTQ files will be QC'd and you can combine them into datasets. All checked-in datasets are available in the datasets overview. You can edit the datasets with your team and add metadata annotations, which you can later use in your data analysis pipelines. You can group associated datasets in projects, which helps you to keep track of your data inventory. On the project overview, you can view and edit all your projects, assign datasets, and build a consistent structure for your metadata. You can also upload related files, such as QC reports, as attachments. ReadStore integrates well in your data analysis workflows. You can use the ReadStore command line interface to access, for instance, file path of your FASTQ files using consistent dataset names. You can also directly connect to the ReadStore database from R or Python code using the open source clients. With this, you can automate and standardize your workflow and get access to your metadata annotations. Efficient management of NGS experiment is a key requirement for research organizations, as it greatly impacts productivity and the quality of the data. And the different perspectives, scientists, for instance, need to have quick access and overview of available data sets. Data scientists need to be able to programmatically access databases that hold these information. Teams need to have a working environment where they can collaborate on individual data sets and the metadata that belong to these data sets and exchange data sets. And then on an organizational perspective, there is the requirement to actually catalog data assets systematically and in a compliant manner, and also foster the collaboration between individual research teams in order to drive future innovation and, for instance, product development. However, in practice, there's still a lot of bottlenecks in managing NGS datasets. Oftentimes, raw and metadata are not consistently stored together since FASTQ files are very large in size. It's very complicated to exchange datasets, and there's no collaborative environment to consistently work on projects in teams. Finally, organizations also oftentimes don't have a consistent repository of data and overview what data assets are actually available. So let's talk about some key features of a data platform that would help us with the problems we just discussed. So first, we would like to have a solution for data management. So a simple user interface that everybody can use through the browser to access all data sets and projects available within our organization. We want to store FASTQ files together with metadata. And on a team level, we would like to be able to collaborate and exchange data sets to have a collaborative workspace where different people can work on individual projects. And we would like to have an integrative command line interface that allows to access the data platform programmatically, and also a SDK or API so that we can access the data platform from R or Python scripts and get the necessary information in order to automate our data analysis pipelines. The ReadStore platform brings together all these features into a single comprehensive solution so that you and your team can focus on actually analyzing data and generating interesting results instead of wasting time with manually curating FASTQ file path and metadata. And with this, I wish you a great day.